Okay, the next and the last scientific speaker for the symposium today is going to be our very own Dr. Nathan Sherer. Nate is currently a professor here in Mercado, and he is a director of the Institute for Molecular Virology and the director of molecular and cellular pharmacology graduate programs. Nate did a PhD at Yale, followed by a postdoc supported by a prestigious EMBO long-term fellowship. And I strongly encourage the trainee in the audience, ask him how to do that because it's really a great scientific journey if you do in two continents. I did that, but I did other way around. So that allowed him to continue to study HIV in Europe at King College London. In medicine, Nate group study virus-host interactions, like imaging and antiviral therapeutic strategies. And today he's gonna share with us his lab's recent work on HIV and cell cycle modulation. Welcome, Nate.
Okay, thanks, Wee, for the introduction, and thanks to Paul and Paul for the invitation to speak. This is a tremendous honor to speak as one of the only non-Temin trainees. And I had a bit of a, got to make sure I figure out how to do this. So we got, okay, this one's forward. I had a bit of a panic attack yesterday, just recognizing that the talks coming before me were going to have such personal connections to Howard, and really being able to talk in a vivid and very meaningful way. And while I can't do that, I was a freshman in college and still an English major when Howard passed. I can at least speak about Howard's legacy here at UW-Madison, or my experience of this over the past 25 years. I first came to McArdle as a prospective graduate student in 1999, and by that point I was a biology major. I had given up on Shelley and Byron, and I had my mentor at Grinnell College in Iowa. Had incredible great fortune of having a virologist, Bruce Boyles, who taught a full-on virology course. And I knew who Howard Temin was, and I knew how important he was. And when I walked into this building during my interview for the microbiology grad program, it felt like a religious experience, and it still does. And it's kind of shocking to think that 12 years later I was trying to teach reverse transcription in the same classroom as Howard. I said to his daughter Sarah last night that it always makes me feel very nervous. We have a whole process of homage that we do to Howard and reminding the students of where we are and what's happening. But I said I always imagine Howard probably rolls over in his grave every time he sees me struggle to get to explain second strand transfer. And but again, it's been an incredible privilege. And the kind of the second aspect of Howard's legacy is my lab occupies Roland Ruckert's old space and Bach Labs in the Institute for Molecular Virology. Famously, let's see, how do I do that? Whoops, that's too soon. How do we do the laser pointer, I guess? Is it, okay, this is it. Okay, so, famously Howard would go for walks and was incredibly invested in communication and maintaining a vibrant virology and genetics and X, Y, and Z community at UW-Madison. So the Temin path has always also been a hallowed spot. I know he would go for walks with Roland and Bill Sugden and others. And Bill said they would eat sandwiches and talk about everything. But that's where some of the best thinking was done. And this is one of my favorite photos of my parents who are falling in love again on the Temin path. It's a special and magical place. And then going to Yale and getting into retrovirology when I joined Walter Motz's lab as a pipsqueak grad student. We had the luxury of being able to go to Cold Spring Harbor retrovirus meeting almost every year, and seeing John and Steve Hughes maintain the tradition of Howard sitting in the front row near the podium and asking, what was the multiplicity of infection, and all these experiences, and it's already been and the grace and collaborative nature and social justice investment, everything I have learned about Howard and his commitment to his family and his community and his trainees has always been an inspiration. And I think that transcends into his relationship with David Baltimore and their ability to cordially share the honor for the discovery of reverse transcriptase. And a sad coincidence that we lost David Baltimore earlier this week that was already discussed. And then one of my favorite things in my office is, I don't know what I'm supposed to be pointing. And okay, so it was when Betty finished cleaning out Howard's belongings and Rayla had claimed all the key things that she wanted to keep, she asked me if I would like some of Howard's books, and I still keep them above my desk, which is just kind of a cool reminder. And then something that Paul Ahlquist has always said at various meetings that I've always found quite profound is that, as investigators, ideally we generate and invest in great science, but we also generate, invest in great people. And that's kind of led to, the other aspect of Howard where, for me, when I got to grad school, I've had a panel of scientific heroes, which many of them are Howard Temin trainees. I could probably say literally, the most 50% of the literature that I loved throughout my training came from this generation of investigators that we're privileged to be with here today. And I didn't put Sandy up here because she went to the dark side and became a DNA virologist. But I've always appreciated your amazing science. I just don't know enough about it. So, all right. So today I'll talk about some of the work in my group. A lot of this has been led by a talented grad student who's soon looking for postdocs, Maddie Bandini, in collaboration with Asi Suzuki and a gifted grad student in his lab, Deval Gon. All right, I'm not good at this. So we're at the last talk, so I don't have to spend too much time presenting how HIV works, but HIV has 14 proteins and a bunch of accessory proteins. TAT and REV are key auxiliary proteins. The virus doesn't grow in the absence of these. They drive transcription and nuclear export. And then these 4 accessory proteins that play really crucial activities in vivo, but at least in some cell culture systems are typically expendable, so they tend to be immunomodulatory. And I'll talk about this long-standing question. So it was earlier said that retroviruses should not kill cells. Only graduate students kill cells. HIV kills cells, at least replicating cells. It induces severe cell cycle arrest. So unlike Howard's passion for RSV, HIV is not a tumor virus. And why exactly it wants to kill cells is still somewhat of an open question that's been studied for several decades. And there are at least 2 activities in the virus that modulate the cell cycle, and the best 2 that are studied are VPR, which you guys heard a little bit about earlier with Kathy's talk, and VIF. The we pretty much understand VIF's day job. It degrades a cytidine deaminase antiviral restriction factor known as APOBEC3G. If that protein gets packaged into virus particles, it can disrupt reverse transcriptase and hypermutate the genome. So the virus needs to get rid of APOBEC quite quickly before it starts making virus particles. And it does so. VIF degrades APOBEC through proteasome-mediated pathway. VPR also operates through proteasome-mediated pathway. And just because I can't spend time on it, it does 800,000 different things, modulates much of the cellular proteome, and it's been a bloody hard thing for the field to study because of its complexity. And I'll just point out that VIF also does targets the B56, otherwise known as R5 family of subunits of the PP2A phosphatase holoenzyme for reasons that are really interesting but not fully understood yet. And I'll bring that up now because we think that might have some relevance to some of the phenotypes that we've generated. So now we go back in time, and 30 years ago, I think after Mike Emmerman had shaved off his mustache, they spent a lot of time studying VPR and demonstrating it had a potent effect on the cell cycle, arresting cells in G2. So if you do flow cytometry, this does not work from Mike's lab, but this is just a demonstration of a typical flow cytometry profile for viruses that are expressing VPR. You get a second peak indicating 2N DNA. And VIF was also demonstrated later in around 2005 to also have the capacity to arrest the cell cycle, seemingly in a G2M transition, based on what was known from flow cytometry. So at this time, kind of the general idea was that both these proteins are operating to target G2M, and why. I think some of my favorite ideas was one that Mike put forward in the 90s, that VPR promotes cytostasticity. So instead of inducing a cell cycle arrest that immediately kills the cell, it'll arrest cells in G2M, and they'll actually be able to live longer and sort of support the infection longer. And some of the data I'll show is very consistent with that concept. And then I bring up Kathy's model, which I still love, that VPR induces cell cycle arrest to produce a cell stress. So a lot of RNA viruses, for example, produce cell stress that allows them to steal ribosomes from cellular transcripts by shutting off host translation. So that's always been a compelling idea that VPR is helping to modulate the cell and through modulating the cell cycle, promote a cell state that is optimal for the virus. And so we wanted to get into this because the limitation of flow cytometry is that you can't really get the dynamics on what's happening with the cells in terms of morphology or dynamics in the cell cycle. And I don't know how well this is showing up, but these are HeLa cells that are expressing tubulin fused to mCherry, so we can monitor the formation of the mitotic spindle. I keep pointing at the wrong thing. And so you can see instances. This is about 40 hours of long-term imaging. And something that my lab is really invested in is kind of these long-term imaging strategies, so we can try to see entire viral life cycles play out. And our cell line of choice, I'll just say, is HeLas, because they don't move. They'll sit there. You can monitor the same cell for 5 days, and that's highly useful, even though it's not the native host of HIV. It at least works as kind of a reasonable model system. And I'll show what we've learned from these movie approaches, that VPR indeed does target the G2M phase in ways that we think are quite interesting. VIF actually induces a metaphase arrest, so it actually allows the cell to move into M phase and stops cell division just prior to chromosomal segregation. And then I'll try to convince you that VPR can actually suppress VIF's effects, and we think that might be one of the reasons that VPR stops things before things get to mitosis. All right, so this started with trying to visualize APOBEC responses to VIF in cells. So I already told you that VIF degrades APOBEC. If you make a virus in cells, so these are cells over, I don't know how many hours, 24 hours. That's about how long it takes to make virus particles in a HeLa cells. And we make a stable cell line in green that's expressing APOBEC. What you can see in this movie, as the cells get infected with HIV, they're expressing a CFP reporter, so we know which cells are infected. And you can see a redistribution of APOBEC to the plasma membrane, consistent with it being packaged into virus particles. At the time we were setting this up, Vinay and Paul Benash were scooping us on all these ideas, but we love them anyway. And then if you express VIF, you can see proteasomal degradation occurring quickly. But what we saw in all these movies was that whenever VIF was present, but not when VIF was absent, and I'll say this is a VPR minus virus, so I'm not going to bring up VPR yet, the cells would round up in an arrest that to us looked like a metaphase arrest. So we thought that was interesting, and this is just kind of a broad movie with an apoptosis marker in the HeLa cells in green. So when the cells round up and become apoptotic here, the cells turn green. And this is only expressing VIF alone, again, in the absence of VPR or other viral factors. So VIF is really good at killing cells. It arrests cells at metaphase was our conclusion. And then we started asking why, or Jordan and Ed in the lab at the time really became obsessed with this, one of those things that I couldn't get them to stop thinking about. And it seemed interesting because, one, for a virus, shouldn't this be a bad idea? You don't want to kill your host unless you have to. And if VIF's, and we also, because we work in a cancer biology department, Taxol is used to treat breast cancer. The idea is to arrest mitosis. So if you can get cancer cells to arrest the mitosis and always apoptose, that's a good thing. So maybe understanding this pathway could be relevant to downstream efforts to inform a cancer therapy. At this time, Ossie Suzuki was joining the lab, which kind of kicked this project into the next level. Ossie is one of the world's experts as a cell biologist and microscopist focused on mitosis. The details that he can glean from a mitotic spindle are absolutely stunning and incredible. So we showed him the phenotype, and fortunately he was excited to get involved in this project. And Ed in my lab kind of handed this off to Deval in Ossie's lab, working with Maddie, and we started looking at more specific what's similar and different between VIF and VPR head-to-head. And these are data indicating that when you express VIF in a cell, it arrests cells in metaphase. Typically a cell only spends about half an hour in metaphase before it divides into two daughter cells. In the presence of VIF, those cells can sit in metaphase, I think, up to almost 2 days. And at the time, they're just sort of pulling back and forth on the chromosomes, but they never divide. I'll show you a movie in a second. And then they almost always die in enterapoptosis. VPR can induce a mitotic arrest, but it's relatively rare, and usually, if it does, it usually proceeds to division. And just to clear the punchlines quickly, we can see this in many different cell types, including T cells. It's not p53 dependent, as was previously reported. Again, I've already said it differs from VPR. There are defects in kinetic core alignment, spindle cell checkpoint, a lot of details I don't have time to get into today in terms of the cell biology. And then I'll just show you what this looks like. So a typical cell division event, we have labeled H2B as a DNA marker in this cell, and GFP tubulin, and it already went. I don't know if I can go back real quick. Just so you can see, boom, about 2 hours, the cells are divided. We know what that looks like. And then it's kind of fun to watch what happens with VIF, where you get a metaphase plate formed. It's beautiful, looks like a spaceship. The chromosomes line up, the mitotic spindle forms, but you never get clear division, and the chromosomes go back and forth. You end up with polar chromosomes. And then these are just some 3D renderings that Ossie did of, this is a typical metaphase plate with the chromosomes all lined up, and the kinetochore is highlighted in red. And I'll skip ahead to, this is what it looks like with VIF. It's pretty messy. Chromosomes are all over the spindle, and we know are moving, apparently are not appropriately attached and divided. So mechanistically, Ossie's and Duvall's labeling strategy, what we think mechanistically so far is that what happens in the presence of VIF is there's a disalignment of the proteins in the astin-SCAP complex that are needed to preferentially balance the pulling forces. So with that imbalance, sometimes a chromosome will get released at metaphase in the presence of VIF. And then we think probably the reason for that is that VIF not only degrades APOBEC, but it degrades this phosphatase regulatory subunit that plays an important role at the kinetochore at this phase. So we spent a lot of time on the cell biology. Does it matter? Is it important? VIF metaphase arrest is conserved. Jorge in the lab has done comparative analysis of VIF alleles that we isolated from a variety of HIV subtypes that we obtained from Viviana Simon. I'll just go through this analysis quick because of time, but these VIF alleles have differential activities in terms of APOBEC degradation. Some of them aren't as potent as wild type, and then they also exhibit differential effects on the cell cycle. Jorge here developed an imaging-based analysis tool where cell rounding could be used as a quick way to get a kind of mid-throughput readout for cell cycle arrest. And so we think currently that this probably correlates with VIF's capacity to degrade B56, which is a question that Maddie's still working on confirming. So years ago, I showed these data to Mike Emmerman, and I asked him if he thought that VIF was interesting in the cell cycle, and he called it it might be a spandrel. And I like this concept where, the spandrel is the decorative element in archways where, it doesn't really perform an important function, but you can use it to make things look fancy, or so kind of an evolutionary hanger-on or something that's not important. Maddie wanted to see what happens in the presence of VPR because VIF is not expressed alone; it's expressed in the presence of VPR, and had the hypothesis that, well, if VPR really does block things at G2M, then you'll never advance to mitosis, so the VIF effects should not be able to manifest. So she set up a really nice system for looking at this. We do HeLa cells. We image the same cells for 5 days. I was kind of blown away that this was possible, but that way you can get multiple rounds of cell division and monitor them over time. And then she engineered CFP expressing reporter viruses that were either lacking VIF and VPR expression, did one or the other or both, did the infection at MOI of less than 1, if Howard was here, and then and this is just demonstrating that we're getting similar production of the viruses. She carefully titers them. The VIF minus ones don't express VIF. The VPR minus ones don't express VPR. They all express GAG and CFP as expected. And then started doing the movies. So what is this gonna look like? And I'm just going to show a VPR movie here. It's quite different than what we saw for VIF. And I'll see if, hopefully you can see the colors. It looks better on the screen down here than it does up there. But what we see is down here, the cells that turn blue are the infected cells. The cells that are down here that are not blue are uninfected cells. So the first thing you'll notice is that the VPR expressing cells are massive. 5 to 6 times the size of uninfected cells in the absence of VPR. The second is that I think this movie is 83 hours, and you did not see a cell division event. for those VPR expressing cells, which we found quite remarkable. So those cells are persisting effectively in a cytostatic state for ages. So then what happens when we look at the actual readouts for VIF and VPR? Time in mitosis, as I showed you before, in the presence of VIF, that can be up to 2 days. In the presence of VPR, there is a bit of delay in mitosis, but it's not as pronounced as VIF. Put them together, it actually goes down. That's interesting. Time in interphase, so these are all the other phases upstream of mitosis. VIF doesn't really affect interphase at all based on our measurements. VPR does because of the G2M arrest. And then we saw this thing that's kind of fascinated us, where if you express VIF, it seems to actually be extending this cytostasticity and VPR's ability to keep the cells alive even longer. So you can get those cells that are alive for 5 days. And our vision here was they continue to pump out virus particles throughout this time. So VPR's arrest is dominant over VIF. The cell never makes it to M phase, cytostatic. And we think that VIF is actually doing something potentially through modulating the phosphatase pathways to help VPR extend the cell lifespan in a way that might be beneficial to the virus. So these are kind of new functions that we've proposed for the proteins. This kind of summarizes what I said before, so I'll move on. The other analysis we did was the Pip-Fucci system, and this is a cool imaging-based system where you can generate cells that are expressing proteins that allow you to delineate the cell cycle using fluorescent protein reporters. And in this system, the CDT1 and venous reporter is expressed during G1. That gets downregulated at the onset of DNA synthesis. And you get upregulation of geminin, which is fused to M-cherry, and then during G2M, both proteins should be expressed. So you can define G2M as both colors active, S phase, geminin only, and G1 as yellow or green in this case. So we did a YFP M-cherry system for this. So what you're going to see in the middle of this movie is the G1 phase. In the red is M-cherry. And these are just happily dividing cells. The cells divide, and you get cells that move from G1, divide, go back to G1, and you can see the S phase pulse. In the VPR cells, it's quite remarkable that you see the cells in G1 phase that moves into G2M, sits there for a while, and then ultimately what we think happens is it reverts back to G1 because we see loss of the S phase marker, and then those cells continue to exist and thrive. Here's the quantitative data, so I'll just point out that this is consistent with this notion. The other thing that happens is, because the cells do go through S phase without breaking down the nuclear membrane or proceeding to mitosis, is they duplicate genomes. So you get polyploidy, which is again consistent with observations from Mike's lab and others, consistent with something that in cell biology is known as endoreplication, wherein the cell is duplicating chromosomes without proceeding to cell division. All right, so what's in it for the virus? I'll go through this very quickly. The last cool thing I just want to emphasize is that one of the things Maddie predicted based on endoreplication is if the chromosomes are duplicating, you just saw Vinay show a nice integration event. If the cells go into S phase but they never divide, they might duplicate the number of integrated proviruses, and that's exactly what we see in the presence of VPR, and I'm in trouble. And that corresponds with a twofold increase in viral gene expression consistent with having 2 genomes instead of 1. And then when you factor in the fact that those cells are not dividing, we see an up to 5-fold increase on a per cell level, both in the HeLa model as well as in T cell lines and in primary T cells, where we see the same effects with endoreplication, duplication of genomes, and an increase in per cell virion output. So this is kind of our working model that VPR helps to suppress VIF-induced cytotoxicity, helps to keep the cells alive long-term, allows the genome to actually duplicate 2 proviral genomes. It's not massive, but it's better than 1. And that kind of brings us to our conclusions. We think that VIF and VPR are working together. We're trying to figure out how important is this for the holy grail in the field, latency reversal, where like if you can inactivate VPR, then when latency reversal happens, can you target the cell cycle to kill the cells? And then is the PP2A by VIF helping to form a preferential cell environment that helps the virus produce genes and variants? So I'll finish just again by acknowledging especially Maddie Mandini, who's done the VIF and VPR work, help from Jim and Jorge in the lab, who are kind of our cell cycle team currently. Aussie's amazing group, especially Duvall and former undergrad Kaylin Stevenson, and my alums Ed, Jordan, and Lorraine. Funding in this age, very grateful to taxpayers that allow us to do these things, and hope that continues, and thank you for your time.
We have time for like 2 questions.
Oh, very nice. You might think, though, in cell culture you get an increase in virus productivity, but you might think that aberrant cells like that would be real good targets for elimination in vivo. Has there been any experimentation done in vivo with mutants that might address in primate models of mutants that would address this sort of issue?
We are trying to set that up personally. So Aussie and I submitted an R1, and it came back with basically, how are you going to test this in vivo? So we've been talking to Priti Kumar, who has a humanized mouse model, where we can go in with those viruses and both look for endoreplication and look for persistence of cells and see if they do have multiple genomes with or without those combinations. Beyond that, all I can say is there are prior reports that some cells isolated from lymph nodes have been shown to be polyploid in this sense, beyond that, don't know. I don't know if Mike has done some at least VPR work in monkeys going back a long time ago.
There's some of those that go way back, and Ron DeRozier,
at least
for
showing
example,
it's conserved and it's important, but yeah.
They're quite defective.
Right. So different model.
It's complicated.
Beautiful. You kept referring to by having the cell hang around for multiple days, you could have large virion production. Have you actually done any experiments to show large virion production?
Yeah, on a personal level, I mean, it's not massive, but I think we see typically 3 to 5-fold more virion output per when you factor in cell number. It gets let's see, I can show it gets a little tricky because in the so this is kind of the demonstration of what you see if you look head to head. If you look at a VIF minus VPR minus cell culture for HeLa, for example, the cells are growing, that's in black, and they're producing more virions over time, that's in blue, based on harvesting the virions and sedimenting and measuring by Western blot. And then you look at the condition for the VPR conditions, you get similar virion output over time, but those cells are not dividing. While in the VIF-only context, if the cells were dying, everything just crashes.
So per cell mass,
for cell mass, I don't know.
That's
an interesting question. We only really see like a 2-fold increase in single cell late-stage gene expression, even in the big massive cells that are expressing VPR. It's not a 10-fold effect, which I was always told is a true virological effect, but we think it's probably enough to help for sure.
Okay, we take one last question.
So I hope you don't mind a question from a non-virologist, but it has to do with the endoreplication. So as you probably know, megakaryocytes are cells in the normal bone marrow that become polyploid and are not destroyed, and it's part of their biology. Are there any similarities in the regulatory mechanisms for the polyploidy of HIV-infected cells and megakaryocytes? Any insights on that?
I have no idea. Yeah. Yeah.
All right, it was worth a shot.
Mike, so you're the other expert.